This zebra and this angelfish, pterophyllum, how do two such completely different species end up with the same pattern? And it's not just those two. This ladybug and that cheetah, this snake and that Dalmatian dog, you'll find thousands of examples like this. But the question remains the same. How is it that such vastly different species display identical patterns? What's the secret behind these patterns? Before we dive into that discussion, please like the video and subscribe to the channel. So we can ask two questions here. First, how are these patterns formed? And second, what is the mystery of the same patterns appearing in different species? The safest answer to the first question is genes. That answer isn't wrong, but it's not the full story. It's incomplete. Why? Well, before we learn that part of the story, we need to know about the hero of this tale, Alan Turing, often called the father of modern computer science. Alan Turing was a genius mathematician. When he saw the same kinds of patterns appearing across various species, he thought, as a mathematician would, that there must be some universal mechanism creating all of them, and there should be a mathematical equation describing that mechanism, an equation in which changing a variable's value produces different patterns. So he set out in search of exactly such an equation. After studying thousands of patterns, he finally devised one and published it in a research paper titled The Chemical Basis of Morphogenesis. And here it is, the reaction-diffusion equation of Turing. I'll come back to this equation at the end of the video, when it will make much more sense. According to Turing, there are two kinds of chemical molecules, activators and inhibitors, which he called morphogens. Right at the start, genes produce very small amounts of activator and inhibitor molecules, and these then spread themselves out over the entire body surface, i.e., the skin. Once the concentration of these morphogens reaches a certain threshold, the system stops producing any more genes or morphogens. The special thing about these morphogens is that the activator increases both its own number and that of the inhibitor, whereas the inhibitor neutralizes the activator and decreases its number. So, the activator molecules made by the genes go on to produce still more activator and inhibitor, which then diffuse across the skin. As the inhibitor diffuses and encounters the activator, it reacts with it, neutralizing or destroying it. Because the inhibitor multiplies more quickly, it forms a ring around each activator center, confining it to a tiny region. And that's how a single spot is created, which is exactly the spot pattern you see on a cheetah. You can think of it like a dog herding sheep into a pen. The dog is the inhibitor, and the sheep are the activator. A key point is that to get spots, the inhibitor's diffusion rate, or how fast it spreads over the skin, must be higher than the activator's. Only then do spots form. If you are still here, please like the video and subscribe to the channel to get science content like this every week. As you can see in these images, at t equals 0, the surface is completely uniform. A little later, at t equals 10, the activator shows up in red and the inhibitor in dark blue. By t equals 100, the inhibitor has spread everywhere and has corralled the activator into tiny isolated spots. If the activator's diffusion rate becomes a bit too high, those molecules start leaking out of the inhibitor's ring, and instead of spots, you get short stripe-like lines of activator, what we call a labyrinth pattern. Now, as I promised, let's look at the equation. In it, Q is an unknown vector-valued function that we must solve for. D is a diagonal matrix whose entries are the diffusion coefficients of the activator and inhibitor molecules. In plain language, D encodes each morphogen's diffusion rate. R is a vector-valued function describing how, given concentrations Q, the morphogens react with one another at each point on the skin. Thus, the first term on the right side of the equation gives us the diffusion behavior, and the second term gives us the reaction behavior. That is how the reaction-diffusion equation, RDE, is constructed. If we change the diffusion coefficients, that is, vary D, we obtain different solutions Q, hence different patterns. Likewise, if we change the shape or area of the surface, the skin, we get different patterns. For example, on a cheetah, the broader torso area yields spots, whereas on the legs or tail, where the skin area is narrower, stripes appear instead of spots. Turing's discovery was decades ahead of its time. Turing's equation is so fundamental and universal that many other natural processes obey similar equations. For instance, there's Fisher's equation for modeling biological populations, the Newell-Whitehead-Sagel equation for Rayleigh-Benard convection, and the Zeldovich-Kamenetsky equation used in combustion theory. Turing's theory doesn't just explain biological patterns, it also accounts for non-living phenomena, the bismuth monolayer pattern, wind-blown ripples on sand dunes, the swirling shapes of flames, even the uneven mass distribution in galactic disks, all can be described by Turing-type reaction-diffusion dynamics. That's the power of his insight. The takeaway is, no matter how random something in nature might appear, no matter how much you feel, it's just the way it is. There's always an underlying scientific mechanism, even if we haven't discovered it yet. Had Alan Turing never proposed this theory, or had we never learned about it, the activator-inhibitor processes would still be shaping patterns in the world. We just wouldn't understand why. After all, everything happens for a reason. Since I alone run this channel, it takes me a lot of time and effort to make these videos. So if you like them, or at least appreciate the effort, please like the video and subscribe to the channel. It means a lot to me. Also, if you'd like to see a video on any other topic, let me know in the comments. Thank you.